So students will talk about here about the nucleic acid. So nucleic acid is the polymer of nucleotide, and nucleotide is made up of nitrogenous base, phosphoric acid, and pentose sugar. We can see here the nucleotide. The components of nucleotide is nitrogenous base, phosphoric acid, and pentose sugar. And if we will talk about the nucleoside, so there is uh, no phosphoric acid in nucleotide. It means if we will remove the phosphoric acid from the nucleotide, then it will be called as nucleoside. So here we will see the structure of a single nucleotide. So nucleotide, as we have already seen, that is made up of sugar, phosphate, nitrogenous base, phosphoric acid, and pentose sugar. So we can see here the S denotes sugar, pentose sugar, and N is nitrogenous base, and P is phosphoric acid. So we can see here the nitrogenous base here. The nitrogenous base is attached to 1 dash carbon of pentose sugar by N glycosidic bond. So we can see here the nuclear nitrogenous base is attached with this sugar with 1 dash carbon through glycosidic bond. However, on the other hand, you can see here that phosphoric acid has been attached with this sugar with 5 dash carbon. So here it has been written that phosphoric acid is attached to 5 dash carbon of pentose sugar by phosphoester bond. So here we can see the different bond. So nucleotide is attached with the sugar through glycosidic bond, and the phosphoric acid through phosphoester bond. So we can see here that one nucleotide is here, and below this another nucleotide is there. So both of these nucleotides are again attached with each other with ester bond. So here in between two sugars, phosphate is present, and there are two phosphoester bonds. That's why these bonds are together called as phosphodiester bonds. So we can say that two different nucleotides are attached with each other through phosphodiester bond. And we can see here 3 dash in and 5 dash in. So here uh, on the sugar side, OH group is there. So OH group is denoted by 3 dash in, because OH group is attached with 3 dash carbon of this sugar, and phosphorus is attached to phosphorus. On the other hand, the phosphorus is present, which is present on the 5 dash end. That's why the another end of the polymer of this nucleotide will be called as 5 dash end. So each and every places when we see the sequence of nucleotide, then one end will be called as 5 dash end, and another end will be called as 3 dash end. So here we will see the details of DNA, with detailed information about the structure of DNA. So it is a polymer of nucleotide, polynucleotide, and these are arranged in a linear fashion. It means the nucleotides will be arranged in a linear fashion, and it will form a polymer which is called as DNA. So here the nucleotide is composed of following chemical substances. So pentose sugar is made up of, since it is made up of 5 carbon, that's why these are called as pentose sugar. And these are in case of DNA, these are deoxyribose. And in case of phosphoric acid, it is common in between DNA and RNA. And in case of nitrogenous base, we see here that 4 types of nitrogenous bases are there in DNA, that is adenine, guanine, cytosine, and thymine. So 4 different types of nitrogenous bases are there in the composition of nucleotides of this DNA. And adenine and guanine are called as purines. However, cytosine and thymine are called as pyrimidines. So we can see here that pyrimidines are bicyclic structure, and on the other hand, pyrimidines are single ring structure. So here we see the Chargaff rule. So Chargaff rule is based on the pairing of pyridines and pyrimidines. He stated that the DNA is composed of equal amount of purines and pyrimidines, because the number of A adenine will be the same as thymine, and the number of cytosine will be the same as guanine. So here we see that the pyrimidine is equal to purine. So the number of pyrimidines in a DNA is equals to purine. So here we see that always T binds with, or we can say make a hydrogen bond with G only, and vice versa, and T always attached with adenine through hydrogen bond, and in between C and G there are three bonds, and in between T and A there are two hydrogen bonds. So we will in further slides we will see the details. So here we see the DNA model proposed by Rosalind Franklin, Wilkins and co-workers. So this group has first time proposed the structure and model of DNA. So in 1940, the X-ray diffraction studies performed by Rosalind Franklin, Wilkins and co-workers revealed the three-dimensional structure of DNA, and as per them, the DNA has the following structure. So we will see here that what they concluded by their X-ray diffraction studies. So it is a helical structure. Second point is it is long and thin with a uniform diameter of 20 angstrom, and the distance between the successive nucleotides is 3.4 angstrom. The fourth point is it is a single helix made up of complete turn of every 34 angstrom, that is 3.4 angstrom. So we can see here the structure of DNA, that this is a helical structure. So helical means helical and double stranded structure. Double stranded means there will be two stranded strands, and both of them will be spirally arranged with each other, and the complete turn, the one strand will complete its one turn with 45 angstrom of length. However, the diameter will be 20 angstrom. So here we will see the DNA model proposed. It means proposed by Watson and Crick and Wilkins. So further, the uh, uh, DNA model was proposed by J.D. Watson and Crick along with Wilkins. So this group again modified, and they did some more experiments, and they proposed this model. So they proposed the double helical model of DNA structure, and according to them the DNA has following structure. So we will see what they concluded, what this group further concluded. So the DNA is made up of two long parallel polynucleotide chain. Both the strands are helically coiled around the same axis. So first point, they concluded that DNA is made up of two parallel nucleotide chains. It means two 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 uh, two strands will be formed by the polymerization of nucleotides, and both the polymers will form a chain kind of structure, and both of them will be arranged helically, and uh, they will make a helix through a same axis. So the second point is the bases of each strand paired together by hydrogen bond to form a right-handed helix. So this helix will be in right-handed form. So we see here that both the strands are attached with each other through hydrogen bond, and purine of one polynucleotide chain means purine of one strand will pair with the pyrimidine of another chain. So the third point is the both the chains of DNA are complementary to each of them. It means one strand will be complementary to another strand, means the second one. The fourth point is the backbone of strand is made up of alternatively arranged sugar and phosphates. So we have seen this structure previously. So there is a, we can see here that in this figure, sugar phosphate, sugar phosphate. So backbone will be formed by uh, alternative arrangement of sugar and phosphate. We can see here, and uh, each and every sugar will be attached with this nitrogenous base. So here we were talking about structure of DNA. Okay, so we were talking about this Watson-Crick model. So the both the chains are complementary to each of them. Fourth point was backbone of strand is made up of alternative arrangement of sugar and phosphate. And the fifth point is both chains are anti-parallel. It means I have already told that each of the chain is uh, denoted by 3 dash and 5 dashing. It means both of them will be arranged in anti-parallel structure. It means if one chain will be in 5 dash to 3 dash direction, then another chain will be in 5 dash to 1 in. If one is arranged in 3 dash to 5 dash, then another will 5 dash to 3 dash. It means both the strands will be in anti-parallel position. So Watson, Crick, Wilkins were awarded with the Nobel Prize for their structure in 1962, and the size of we will see some the genome size of some organism. So lambda bacteriophage is having 48502 base pair. However, in case of E. coli, a bacteria is having 4.6 into 10 to the power 6 base pairs, and in case of human, it is 6.6 into 10 to the power 9 base pairs. So these are the total amount of uh, base pairs of means base pairs means the pair of nucleotides. We see here the here note. Sometimes the DNA is also present in sometimes DNA is also present in single stranded structure. So in some of the viruses, the DNA is also present in single stranded form. So we can see again the structure of DNA proposed by Watson and Crick. So we can see here there are two strands. One is in 5 dash to 3 dash direction, and another one is uh, 3 dash to 5 dash, and it completes a single turn in 34 angstrom, and the diameter is 20 angstrom, and uh, in a complete one turn it is having 10 base pair per turn. So we will see here the structure of RNA. So again, RNA is made up of polymers of nucleotide. So, but here some basic difference will be there as compared to DNA. So we will see here the again its nucleotide is made up of nitrogenous base, phosphoric acid, and pentose sugar. But the basic difference is here that the nitrogenous base of RNA will have uracil in place of thymine in DNA. So all the nitrogenous bases will be same in RNA, but in case of this particular nucleotide, uracil will be there in RNA. In when, however, in case of DNA, there will be thymine in place of uracil. And in case of pentose sugar, we will see here at the basic difference. The DNA's pentose sugar is made up of deoxyribose sugar. However, here in case of RNA, the pentose sugar is ribose sugar. So U is present instead of T present in DNA. So types of RNA, we will see the types of DNA. So type of RNA. So there are three types of RNA. One is ribosomal RNA, that is also in short called as rRNA, and it is about 80% of the total RNA. So the second one is messenger RNA. It is also called as in short mRNA. It is in 15%, and the transfer RNA. In short, it is called as tRNA also, and it is very less amount of RNA out of total. So it is of 5%, and both of all these three types of RNA is differ uh, on the basis of their size and their function. So all these three RNAs have different structure, size, and function. So first we will talk about rRNA, that is ribosomal RNA. So these uh, rRNA are synthesized in nucleolus. First thing. The second point is it is structural and functional component of ribosome. It means ribosome is made up of rRNA and protein. The second point is it is most abundant, that is 80% of all RNAs, and it is single-stranded, most stable form of RNA, and rRNA molecules bind to mRNA and tRNA molecule, and it is present in cytoplasm of prokaryotes and eukaryotes. So we have seen some details of rRNA. So basically, we should know that it is the basic component of ribosome. The second RNA is mRNA, messenger RNA. So these are produced in nucleus. We can see the difference here. The rRNA is made up of is synthesized in nucleolus. However, in case of mRNA, it is produced in nucleus and it carries information for the synthesis of proteins. So what happens? It it transfers the genetic information from DNA and helps in the synthesis of a particular protein. The second point is it is specific mRNA synthesize specific protein. So each and every protein will have their own mRNA. So the next point is very few in number, that is less, that is very less amongst all the RNA, that is 5%, and it is single stranded, ranges from less it is ranges uh, from less to very high number of nucleotides, and it's uh, when we will see the stability of this mRNA, then we will see that some of the mRNAs have very less stability and some will have more stability. So there is a wide variation. So here we will see the mRNA is the exactly complementary copy of the segment of DNA. It means the mRNA will be complementary of the DNA. So uh, we see here the basic difference between okay. So we will see here how the complementary, how the mRNA will make the complementary. When A adenine is present in DNA, then in case of RNA, always U will be present. If T is present in DNA, then always its complementary A will present in RNA. If G is present in DNA, then C will present in RNA, and if C is present in DNA, then G will present in RNA. So we can see here the RNA how it makes the complementary of the DNA. So what happens? This mRNA always carry the details of genetic information to the cytoplasm, where by the help of ribosome and rRNA and tRNA, it helps for the formation of or synthesis of protein or specific protein. So we will see here the mRNA can be divided into two main categories. One is monocystonic mRNA and another one is polycystonic mRNA. So monocystonic mRNAs are those mRNA which encodes a single polypeptide. It means a single mRNA will form a single polypeptide. It always present in eukaryotes, but in case of prokaryotes, polycystonic mRNA is present. It means a single mRNA can encode two or more than two polypeptides. So we can see different categories of this mRNA, and we will see here the difference between prokaryotic and eukaryotic mRNA. So some characteristics we will see here, that in case of prokaryotes, the nature of uh, this uh, difference between prokaryote okay, so here we can see In case of prokaryotes, the mRNA is polycystonic, but in case of eukaryotes, it is monocystonic. In case of prokaryotes, mRNA is less stable, but in case of this eukaryote, the mRNA is highly stable. And non-coding region in mRNA is absent in prokaryotes, but non-coding region is present in eukaryote. The next point is post-transcriptional processing. In case of prokaryotes, it is not required, but in case of eukaryotes, it is required. And 5-methyl guanosine capping is absent in case of prokaryotes, but present in case of eukaryotes, and polyadenylation is absent in prokaryotes and present in eukaryotes. Some of the points are represented here in difference in these differences will not be able to uh, means you people will not be able to understand, but in some further videos we will see the details, then you will be able to understand what is the meaning of 5 dash 5 methyl guanosine capping and polyadenylate formation in non-coding region in mRNA. So further in further videos, uh, I will explain it. So here we will talk about tRNA. So tRNA, that is also called as adapter molecule or transfer RNA. So these are produced in nucleus. So we can see the difference between tRNA, mRNA and rRNA. So rRNA is produced in nucleolus. However, mRNA and tRNA are produced in nucleus. So these are produced in nucleus and transfer. So we will talk here about its function. Transfer scattered uh, scattered amino acids in cytoplasm to their respective codons present in mRNA. So what it does, uh, tRNA transfers amino acid which is scattered in the cytoplasm to mRNA. So it helps to incorporate amino acid to polypeptide chain. So here we will see the size of tRNA. Size is about 80 nucleotides and it is the smallest RNA. The it forms a secondary structure called its secondary structure is called as clover leaf model, and it is formed by the hydrogen bond. And the 3D structure is called as inverted L structure. So here we see the detailed structure of tRNA. So here we see that this tRNA is having 3 arms. So the left arm is called as amino acyl synthetase binding loop. However, on the right it is called as a ribosome binding loop, and the middle one is called as anticodon loop, and on the 3 dash end of this RNA, that is tRNA, ACC nucleotide is present. It means it is conserved, and this is the region where ECC means the amino acid is get attached to this 3 dash end. So when we will see or talk about the translation, then we will be able to understand the function detailed function of the these arms. So we will see here the anticodon present on the terminal end of the tRNA makes complementary binding with codon mRNA. For example, CGA anticodon binds with CGU of mRNA. So this anticodon will be complementary to the mRNA. And the second point is at 3 dash end. Here we can see at the 3 dash end CCA nucleotides are present at the end binds with respective amino acid according to anticodon present on it. It means at 3 dash end different nucleotides will bind, which is basically based upon the sequence of anticodon, and this anticodon will be complementary to the sequence of mRNA. Next point is an enzyme amino acid tRNA synthesis. tRNA, it is also called as T ligase, catalyzes the attachment of a given amino acid at 3' end of tRNA. there are 20 enzymes each for the respective amino acids. So as we know that there are 20 types of amino acids, so 20 different types of enzymes are there to attach these amino acids to tRNA. So amino-acyl tRNA synthetase, or it is called as tRNA ligase, which catalyze the attachment of amino acid to 3' end of tRNA. It means different amino acids will get bind to this tRNA by this basic enzyme that is called as amino-acyl tRNA synthetase. So the next point is one arm of rRNA binds with enzyme amino-acyl tRNA synthase, and which activates the amino acid, and another arm binds with ribosome which hold the tRNA to the ribosome. So we have seen the detailed structure of this tRNA, and to know the function of this tRNA, we will see our next video where we will see the function of these tRNA, mRNA, and rRNA. So now we will see some another RNAs, different RNAs. One is small nuclear RNA, snRNA. So these are present in nucleus, smaller in size, helps in pre-RNA processing, and it is found in eukaryotes only. So we should know that when transcription happens, it means DNA encodes mRNA, then the mRNA will be formed in the form of nascent mRNA, and that nascent mRNA will have coding and no non-coding sequences. So here we see that it is present in nucleus and smaller in size, helps in pre-RNA processing. So uh, most of the RNA, when these are produced, will be present in pre-mRNA or nascent RNA, but after processing it gets more mature. So these will be involved too in the processing of pre-rRNA. Another one is hnRNA, heteronuclear RNA. These are large pre-mRNAs made by DNA polymerase II. It is processed in nucleus to become cytoplasmic mRNAs. As I told that most of the RNA are produced in their nascent form, that is called as pre-mRNA. But for the formation of mature mRNA, some processing happens. So before processing, the mRNA means unprocessed or unmatured mRNA will be called as hnRNA or heteronuclear RNA. So here we will see the difference between DNA and RNA. So first difference is based on sugar. So in case of DNA, deoxyribose sugar is present, but in case of RNA, ribose sugar is present. The second point is genetic material. So DNA is the genetic material, but most of the time, except some viruses, that is retroviruses, uh, RNA is not a genetic material. Third point is strands. DNA is double-stranded structure, however, RNA is single-stranded structure. Fourth point is molecular weight. The DNA's molecular weight is high as compared to RNA. Next point is the nitrogenous base. The DNA's nitrogenous base is ATGC, adenine, thymine, guanine, and cytosine. But in case of RNA, there is uracil in place of thymine. The next point is Chargaff rule. So DNA always follow Chargaff rule. However, RNA doesn't follow. The next point is replication. DNA, in case of DNA, replication occurs, but in case of RNA, doesn't occur. Replication means formation of their replica copy. It means the increment of their number. The next point is helical structure. In case of DNA, the DNA is present in the helical structure. However, in RNA, we have seen that DNA such kind of structure is absent in RNA. Thanks.